All right. Hello, everyone. Welcome. We at Contactica are here with this seminar for ReBIOlution. We have Alvaro and Jaime here as well, and I'm Dario. We're going to wait a couple of minutes to see if some more people join because there were, I think, 19 people registered, and then we will start. All right. Welcome, everyone, to this webinar for the ReBIOlution project. Breaking down microplastics from science to solutions. And we have 1 hour to hear two leading researchers in the field. We have Dr. Patrizia and Marieke, and they are gonna talk to us a little bit about the fragmentation of certified soil biodegradable mulch films and the cutting-edge modeling on microplastic accumulation and exposure. So if you're ready, Patricia, you can take it away.
Okay, let me share my screen. Okay, I hope you can see everything well right now.
Yes,
so,
we can. Yes.
Perfect.
Great.
So thanks again for the introduction and also for the invitation to present today at the ReBIOlution webinar. My name is Patrizia Pfohl. I'm leading the micro and nanoplastic lab at BASF in Ludwigshafen. And today I would like to talk to you about the fragmentation of soil biodegradable mulch film and what is actually the difference between fragments that are released from such certified biodegradable products and from persistent microplastics. So in the focus today, we have our certified soil biodegradable EcoVio M2351. This is a biodegradable polymer blend, which is used for the production of mulch films, and those can be used in agriculture to increase the crop yield. To obtain a certificate for biodegradation of this product, we need to conduct biodegradation tests in soil according to standardized conditions. In this case, it's ISO 17556. And during these tests, we need to make sure that we achieve 90% biodegradation within a given time frame. And as you can see here on the biodegradation curve, we reached the 90% biodegradation already after 181 days. And so we got the certificate for this biodegradable mulch film. But then the question came up, it's nice that you achieved the 90% of biodegradation, but what happens to the rest? What is it? Is it something persistent that does not biodegrade? And so in the past, we put a lot of effort in understanding the biodegradation process and also in understanding what these remaining 10% could be. And we had two approaches here to find out more about this biodegradation process and how the polymer carbon is converted into CO2. On the left side, you can see that we worked with 13C labeling. So we had our biodegradable mulch film that consists of different parts, different monomers, and all of these monomers were labeled with 13C separately. And so we could follow the conversion of all of these components into CO2 separately. That was the first thing we could show. And in addition to that, we also had a cooperation with the ETH in Zurich to find out more about this conversion process. And here we also used the 13C labeling and worked in addition with the so-called nanosims technique, which is an imaging technique that can show where the 13C carbon ends up. And as an overview, you have this picture here on the right side. In green is the biodegradable mulch film, and it's shown in green because this is somehow the basis concentration of the 13C label. And then we have these linear structures on top of the biodegradable mulch film. These are fungi that form, that colonize the surface of the biodegradable mulch film during the biodegradation process. And you can see that these linear fungi structures are shown in yellow and red. And this actually means that there is an accumulation of the 13C carbon in these fungi structures. So this was actually the first proof showing that the fungi and the microbes are actually taking up the polymer carbon, using it for building up their cell walls. And so part of the polymer carbon is not converted into CO2, but remains in the soil as biomass. But then another question came up also about the remaining 10% after biodegradation. The microplastic community was asking if these remaining 10% could also be at least partially persistent microplastics. And so also in this case, we wanted to find out if that could be the case. But since I don't know how much you actually know about microplastic, I would like to quickly show you an overview of what microplastics are and how they are defined. So microplastics are small particles that are smaller than 5 millimeters in size. They are insoluble in water and consist of solid polymers. There are actually two types of microplastics. So first we have primary microplastics, so microplastics that are already smaller than 5 millimeters from the beginning of their life cycle. You might know them as ingredients in cosmetic products or also in the past in toothpaste, but these are now regulated and they only make up around 1% of the overall microplastics. Then the main source of microplastics in the environment are actually secondary microplastics. So microplastics that are formed from macroplastic products due to degradation of macroplastics in the environment or due to abrasion. So very common examples here are tire abrasion or mismanaged plastic waste. And this whole microplastic issue comes from the fact that these particles are generated from non-biodegradable polymers, and so they persist and accumulate in the environment. So when we look now at these fragmentation and microplastic formation products in the case of mulch films, we see on top here the fragmentation process of non-biodegradable conventional mulch films, like, for example, LDPE. And here we have the main fragmentation pathway via UV aging or mechanical abrasion. And these fragments that are formed due to these processes from the conventional mulch films are non-biodegradable, and this is why they would persist in the soil. In contrast to that, when we look at our certified soil biodegradable products, we also observe fragmentation. And this is because of the biodegradation. So when a material biodegrades in the environment, it needs to get more brittle. It gets to be smaller and smaller, so that we actually form degradation products that can then taken up by the microbes and converted into biomass, water, and CO2. And so the big difference here is that these fragments that are formed from the biodegradable products are not persisting in the environment. And to show that this hypothesis actually holds true, we developed a method that enables us to extract fragments from such biodegradable mulch film products from complex environmental matrices. In this case, the matrix is soil. Today, I'm not talking about the process in very much detail, but if you're interested in how we do this, we published the method last year in Method X, and you can look the details up there. So basically, we work with a combination of deagglomeration and centrifugation to get rid of a high amount of soil matrix, while at the same time extracting the fragments from the soil biodegradable mulch film. Very important when we are working with these extraction techniques is that we need to make sure that our fragments from the biodegradable mulch film remain stable during the extraction. So we need to make sure that we do not change the particle size distribution and the total number of particles in the soil, and also that we are not changing, that we are not introducing any changes to the molar mass of these materials. We also need to make sure that we get rid of a high amount of the soil matrix to enable our analysis via IR microscopy in the end. And we need to make sure that we have a high recovery, which we achieved of more than 90%. After extraction of such fragments from soil, we are working with IR microscopy because in this way we can determine every single particle on the filter, determine the identity of every single particle, and also the particle size distribution and number. We are looking at fragments between with sizes between 25 and 1,000 micrometers in this study. So in our actual study, we conducted then a biodegradation test according to ISO 17556 in soil of our biodegradable mulch film. So what we did here was we monitored the CO2 evolution over time of our biodegradable mulch film. We also had cellulose as positive control in the test and LDPE as negative control in the test. And also in this biodegradation test, we achieved 90% of biodegradation of our EcoView already after approximately 180 days. We did not see any biodegradation of the LDPE, and we also saw the biodegradation of our positive control, the cellulose. And then we took out samples at specific time points of the biodegradation experiment. So the first time point was directly after mixing the material into the soil. Then we got another sample after 10% of biodegradation, 30% of biodegradation, 60% of biodegradation, and 90% of biodegradation when we finished the standardized testing. And so after all of these time points, we extracted the remaining fragments from the biodegradable mulch film and applied them onto these filters. And you can see here already that visually that changes are happening to the fragments of the mulch film. So in the beginning, we have quite large fragments, but with increasing biodegradation time, the particles get smaller and smaller. And also we see less particles visually on the filter at the end of the biodegradation test. But then when we look at the microscopy results, so with the IR microscopy, I mentioned it before, we can determine also the identity of every single particle on the filter. And so here you can see the results of only the particles that were identified as EcoBio mulch film at each time point of the biodegradation test. So we look here at the total particle counts over time. And as you can see, we started already with quite a high number of particles per gram soil. But then we actually see an increase of the total particle counts over biodegradation time. And this is because of these fragmentation process that is going on during biodegradation. So the material falls apart, all the particles get smaller and smaller because of the increasing brittleness. And as I mentioned before, this is a process that cannot be avoided and that is also needed to enable the conversion of the material into CO2 by the microorganisms in the soil. And what we then also see when we look at the later time points is that these fragments that are previously formed are not persistent, and also the total particle counts go down over biodegradation time. And if we look at the mass, this reflects this also very well, because this the total particle mass over biodegradation time goes down with increasing biodegradation. So you can see here that it's very essential to actually understand these coupled processes of degradation and fragmentation to understand what happens to the mulch film. What you can also see here is that at the end of our standardized biodegradation test, we still found some particles that were identified as the mulch film after 90% of CO2. But these were less than 2% of the initial mass, so not the 10% that were missing. And we also found that these fragments still showed the same chemical composition as the original fragments. And so we wanted to learn more about how long it would take until these remaining fragments would also be completely degraded into CO2. And this is why we worked together with our partners at the UK Centre for Ecology and Hydrology, since they developed a model for fragmentation of microplastics in the environment. So when you're interested in the model, you can check out the QR code here. And our modelers are Sam Harrison and Kansu Olusegun. And so what they did were they were comparing our experimentally measured particle counts and sizes, and compared it to the modeled data. So you can see here all the time points of the biodegradation experiments. All the dots are actually our measured data, and the bar plots is what the model predicts. The model can look at all the different size classes, also going down to the nanometer size range. And part of the model also includes the conversion of particles into dissolved non-particulate species, and also the conversion into CO2. Here on the right side, you can also compare how the modeled CO2 curve looks like compared to our measured CO2 curve. And so we were quite happy to see that the modeling results here fit very well to our measured results. And so in the next step, we wanted to actually answer the question on how long it would take until all the particles are completely degraded. And with the model now, we can not only look at these specific time points that we also measured, but we get continuous particle size distribution of all the different size classes, and also going down below the 25 micrometers that we are currently not covering with our experimental data. And so finally, we were able to define the time point at which we have less than 1 particle per gram soil in this biodegradation experiment, and this was achieved after 686 days. But as a reminder, again, we have quite a high number of particles that we spiked initially into the compost, and the model also covers all the nanoplastic particles. So now I'm already coming to the end of my presentation. I hope I could get you a good overview of what we are doing here in terms of understanding the fragmentation of biodegradable polymers and comparing fragments of these particles to persistent microplastics. So for us, it's very important that we have this method with which we are able to extract these fragments from compost to make sure that we are not inducing any changes in the particle size distribution and chemical properties of these materials, because then we are actually able to determine the total particle number, shape, size, and chemical composition of such fragments that are released during biodegradation. And I would highlight again that it's very important to understand these coupled processes of biodegradation and fragmentation, and that also a high number of particles does not automatically mean that we have a high mass of polymer in our samples. And we need to understand where these fragments are coming from and how long they would be stable in the environment, and at which time point they are completely converted into CO2. Even though we saw some fragments still at the end of our biodegradation experiment, we can use such models to predict how long they would be stable. And also Marieke will soon tell you a bit more about that on their model, which is also quite nice and looks at these aspects from a different perspective. And so with all of this data, we can, in the end, differentiate how fragments of such biodegradable polymers behave compared to persistent microplastics. So thanks for your attention, and now feel free to ask any questions.
Thank you so much, Patrizia. Yes, we will have some time for questions at the end as well, but if anyone has a question now, you can either write it in the chat and I will read it out loud, or you can just raise your hand and ask it. Is there anyone with a question? In case someone is writing, I will ask one, because I thought it was very interesting, Patrizia, but I was wondering if you've measured any changes in the microbial communities in soil when they are degrading this plastic mulch.
So in this study, we did not look into that, but maybe Kai has some more information on that, if such measurements are ongoing at BASF, or maybe in the past we looked into that.
Mm-hmm. All right. That would be interesting to see, because as well you showed that the half time was pretty long. So like probably it would be, if it was used in the field, it would be covered. The soil would be covered again before all of the particles were gone, right? So it would be interesting to look at that. But
yeah,
super interesting.
Yeah, actually, I heard about field studies. For where these mulch films were applied to for several years, and there are actually degraders that introduce faster degradation than of these mulch films.
All right, thank you. We have a question as well from Ed. who's asking, what happens when you mix lignin in EcoBio? Does that change the assessment of biodegradability? I don't know if you can cover that.
So I'm not sure if this was ever done. I don't think so, but maybe Guy, you know more about this.
Yeah, I mean, lignin is a natural substance with very slow biodegradability, part of the wood structure. There are applications, by the way, that contain lignin, a combination of PBAT and lignin, for example, but these are typically not done to achieve specific biodegradability performance in applications where biodegradability is the key. But in terms of adding natural substances to a biodegradable polymer, what can I say? If you have unmodified natural substances, it does not alter the assessment of the biodegradability of the overall system. So the PBAT, for example, that would be a component of it, shows the full biodegradation as determined in the relevant norms. And the addition of lignin to such kind of a compound does not alter the assessment regarding whether the overall system is biodegradable or not.
All right. I hope that answers you, Ed. If there are any more questions, you can write in the chat and we'll take them after. But for now, thank you, Patrizia, and we can move on to Marieke's presentation.
Thank you. I will share my screen as well. Yes. I think you see my slides. Is that correct?
Yes, we can.
So, my name is Marieke Brouwer, and I work at Wageningen University and research, trying to get a grip on the plastic system and how we can make new products that are more sustainable and less environmentally, with less environmental issues. And today I will tell you something about a predictive model that my team and me made to assess the microplastic accumulation in the natural environment. So I don't know your background, so I will start at the very beginning of why we are doing this. So plastics, they are very useful in a modern society. For instance, in packaging, it makes sure that products can have a very long shelf life, that all kinds of foods can be produced, products can be transported easily, efficiently with low weight, and food products can be consumed. And on the other hand, we see that these plastics in these kind of applications, they have environmental challenges. For instance, the greenhouse gas emissions that are related to the production and use of these plastic packaging, but also the pollution of the environment by mismanaged plastic waste, plastics leaking towards the environment. And when these traditional plastics they enter the environment, they will be persistent and accumulate over time. So we wanted to know what we can do to mitigate this issue. And we have done a lot of studies, and we have found out that we cannot completely prevent plastics leaking towards the environment, especially in these short-lived products in which they are used intensively. A small part of leakage will always be the case. And also products will wear out, there will be abrasion, items get lost. So we need, there's an inevitable amount of microplastics entering the natural environment. And therefore we should include these issues into environmental decision making. So in most current LCA studies, so the typical way of measuring environmental impact, these issues are not included, and that's an issue, because we need to take these issues into account. And therefore, we wanted to know what the quantity of the microplastics is that we are exposed to due to the leakage of plastics in the natural environment, and how this accumulation takes place compared to traditional plastics and biodegradable plastics. So what we found in literature is that there are already a few efforts on trying to get this persistence and biodegradation relation for specific products. But what you see is that they are either based on surface degradation rates, in which the actual chemical conversion from the plastic to CO2 is not included. So these kind of models are not really suitable for the biodegradable polymers, in which we actually want the polymer to break down into CO2 and water. On the other hand, there are models that include these chemical transformations, but they are not suitable for fossil plastics, because they are too limited to handle the really slow degrading polymers. And what we want to do is actually compare what happens if we make products from either a traditional plastic that's not biodegradable or a biodegradable plastic, and how these microplastics emissions and accumulation will relate to each other. So therefore we have started to build a model, and the conceptual basis of this model is that all polymers in the end biodegrade to some extent. Which means that this process can be a few days or months, or it can take thousands of years, but all polymers have some activity in the sense of that they're producing CO2 over time due to a biodegradation process. And what we can do is we can take that as a given and make a mass balance of the polymer substances over time. So we make a mass balance from the microplastics to small intermediate products and to the CO2 that is produced over time. And you will see that some substances are they're more in the beginning, and some substances are more in the end. And with this mass balance, we can calculate the exposure to microplastics over time. And actually also give it a number, so we can for instance calculate the accumulation potential of different polymers and compare that. And this graph will be different for different polymers, for different objects and different environments. So I'm not going into details on all the mathematics that we did to build this model, but just to give you a little bit of background, we built a model based on a set of ordinary differential equations in time, and we fit the CO2 evolution curves that you can measure, and Patrizia already showed as well in a previous presentation. We fit those curves to our model, and with that process we tested multiple variants of a model for both non-biodegradable polymers and biodegradable polymers, and in the end selected the one that was best suitable to model all kinds of polymers. And with the chosen model, we have done this fitting process of CO2 evolution curves and select the best fits, which gives us the data that we're working with. If you want to know more, you can look at the publication. So what we have now is a model that describes the behavior of polymers and it's of the biodegradation behavior of the polymers. And actually, it fits quite well with what we already know about biodegradation processes. So this model, it includes the full conversion from a polymer to CO2 via the depolymerization by enzymes or by hydrolysis, to the utilization of small intermediate products by microorganisms, and in the end, the CO2 that is produced. And this model is suitable for all types of polymers, slow and fast degrading. And now we can start to use this model, and we have been doing that. And we can use the model to, for instance, calculate an accumulation potential as a specific number for a polymer, as some kind of a polymer characteristic. But we can also model the exposure of microplastics when a specific product with specific dimensions, specific material enters the natural environment. Or we can even model what happens when this process is continuously happening, so continuous stream of plastics is leaking towards the environment. Or in case of, for instance, agricultural products, such as the mulch film, what happens if products are seasonally applied in specific contexts. So we can do this, and in the end, this kind of data would be very suitable to be implemented in life cycle assessments to include the impact of microplastic pollution into these kind of assessments. So currently we are working on a database in which we are adding all kinds of different polymers, both slow and fast degrading polymers or biodegradable polymers, into a database from all kinds of mineralization curves that are published in literature. And we can use this to build a background to feed these sustainability assessments. And we can model different polymers, different polymer blends in different environments, of course, and under different conditions. Because, of course, in different conditions or in different environments, a specific polymer can behave differently. For now, we should note that these are examples of specific polymers, and that they are just based on that we can find on literature. So it's not a full database, let's say. And for instance, the blends that we have now implemented in this model, they have, of course, a specific composition. They have a specific background. And furthermore, when we are comparing this data, we should, of course, be careful to check that the conditions are more or less similar in which they are measured. However, this already gives us the opportunity to model all kinds of scenarios of what's happening when plastics entering the environment. So when we are building these case studies and we are assessing these scenarios, they are always built in more or less the same way, which is a similar setup as is done in life cycle assessment studies. And we start with calculating the amount of plastics that is entering the environment. From there, we model what happens to the polymer, and we get the result in the form of a graph of the microplastic exposure over time and the accumulation over time. And in all the case studies that I will show you today, we assume that the shape and the dimensions of the products are the same. But of course, the materials have different material densities. So therefore, the weight in the next slides will differ slightly between the different polymers. So to give you a very simple example of what we can model is the case of a candy wrapper that will enter the natural environment. So it's littered, and it's just one candy wrapper that's entering a soil environment. And here you can very clearly see that the biodegradable materials, the wrappers, will biodegrade, while the non-biodegradable polymers, they will actually persist, and there's no, let's say, real decrease of the microplastics over time, or at least not within the first year. And you can also see that we can model this. So this is a specific example and where we modeled it for specific polymers, but also for a polymer blend. And that for this kind of model, that doesn't, that's no problem. We can do both. And we can also show what happens in a different environment. Of course, this is based on different data because there's different data in literature for the marine environment. But it already shows you that also here the biodegradable polymers, they biodegrade. And and other polymers biodegrade. The biodegradation is much less or none. Maybe also good to explain on these type of graphs is that the modeling, of course, has some uncertainty, and it's based on a database of all kinds of data. So that means that in some cases we have multiple data for instance, in the PBS case, you can see it very clearly here. We had multiple data, and the one data gives a different result than another. So in these figures, you can see the bandwidth of the different results that were found in literature and were modeled. But of course, in reality, littering or leakage towards the environment is not a one-time thing. It is something that happens continuously over time. So it's more important to look at what happens to these polymers once this process is continuing. And here you can see what happens if, for instance, these candy wrappers that we were talking about are littered over time. And therefore, we take the amount of candy wrappers that is available in the Netherlands, is used in the Netherlands, and we multiplied it by a littering rate. And you see that if you then look at different polymers, that the non-biodegradable polymers, they will accumulate quite quickly. Even with small amount of littering, they will accumulate quite quickly, while the biodegradable polymers, they will either not accumulate or they will accumulate very slightly. And this is depending on the balance between the littering frequency and the biodegradation rate of the polymer. We can also check out what happens when the littering rate increases. So in this case, you see we only chose to show two types of polymers here, because otherwise the graph will not be really visible anymore. But you see that the effect for the non-biodegradable polymer when the littering rate increases is much worse than the effect on the biodegradable polymer. What we can also model is what happens if these littering rates are exacerbated. So the EU, for instance, is hesitant to accept biodegradable material for packaging due to a fear of license to littering. And here you can see what happens when we apply very high littering rates for the biodegradable polymers compared to slightly higher littering rates to the non-biodegradable polymer. So for the biodegradable polymer, we go up to 50% littering compared to 1% littering or 3% littering rate for the LDPE. And as you can see, the effect of the higher littering rates for non-biodegradable polymer, it's quickly worse. So you can see the difference for 1 to 3%. It's already a very big difference. While for the biodegradable polymers, the higher littering rates still provide less microplastics exposure than the non-biodegradable polymers with low littering rates. So that's the graph you see in the at the lower end of the slide. And this shows that when you already induce a slightly higher littering rate, biodegradable packaging can limit the exposure to microplastics in the long run. So we also checked out what happens when you have processes that are not continuously littering, but you specifically use a product in a seasonal application, for instance, an agricultural application. And in this case, we took the mulch film as a nice example. And we compare the different materials for different for a mulch film. And you can also directly see in a case that all polymers are recovered from the field, that the mulch film of the non-biodegradable polymer, the material quickly accumulates in the soil, while for the non-biodegradable, for the biodegradable polymers, you see that there's of course these peaks of of microplastics as well, but they die down. And we also wanted to know what happens if we compare this process of an LDPE that you leave on the land, but you recover over time. Of course, you cannot get everything back, so we assumed a 59% recovery rate, to a mulch film that you leave on the land and keep there to decompose. And here you can see that there will be peaks of microplastics on the land in the case of the biodegradable polymer. But in the long run, the accumulation of the conventional plastic is much worse, even though it's mostly recovered from the field. While the biodegradable polymer, in some cases even almost gives a net zero microplastic emission. So this clearly shows the trade-off of the short-term and the long-term exposure to microplastics in such cases. So what we have here to conclude is that we have a model in which we can use to predict the exposure and the accumulation of microplastics. It's very suitable for different polymers, slow and fast degrading polymers. It can be used in assessing scenarios of plastic use and leakage to the environment. And you can use this model to determine the required biodegradation behavior for specific products or applications. And also it can be used to as input in LCA studies. But in those cases, we would also need to understand the effect of these different types of microplastics on the environment and our human health to get to characterization factors of these plastics. Also, these kind of scenarios already show us that biodegradable polymers can be a solution to minimizing the exposure to microplastics over a long time. However, there are some trade-offs that need to be taken into account. For instance, the first trade-off is that there's a difference between short-term exposure and peaks of microplastics that will quickly diminish again, and long-term exposure to microplastics that will accumulate over time. And secondly, in specific product, there's a specific functionality needed. So we need to see how the biodegradation speed can be tuned for specific applications. And if we know this, we can use this model to determine the required biodegradation behavior to limit microplastics accumulation, and therewith hopefully facilitate the transition to a system in which products are designed based on their intended functionality and anticipated end of life, and get to a more safe and sustainable plastic system. This was my presentation. Thanks, and if you have any questions, then please let me know.
Thank you so much, Marieke. That was great. I don't know so much about LCA, but I know that we have some people in Contactica that do, and they have definitely mentioned that modeling for biodegradable materials is super important. So this looks really interesting. If there are any questions from the other audience members, please raise your hand or put it in the chat and I will read it. But yeah, I thought it was very interesting as well. I can start with one question as well, if you don't mind. Because do you think that the rates of like littering will increase if plastics are like biodegradable?
I know that there's a large fear.
Yeah.
And which is fear of that's happening, and that limits, let's say, the process of actually applying these plastics to products where they can have a beneficial effect. I'm not sure that the type of plastic will actually make consumers how do you say, throw away plastics into the environment. How do you say? I think
litter, I
it's a
guess.
matter, yeah, to litter it on purpose. I think that's good. I don't think that it's. So what you see is that there have been studies done on how the littering of different types of plastics, and it's mostly based on the design, on things that trigger people to behave more sustainably with plastics, but the other way around, that plastic, because it's biodegradable, will be thrown away in the environment. I don't know. I don't think that there are studies that show that that's happening. But even if it would happen, I think the biodegradable plastics still are a good option to explore, as you can see by the results of this model.
All right. Thank you for the answer. I think probably still good, but worth wondering. Is there any other questions from the audience? We have some time left for questions, both to Marieke and to Patrizia. Yeah, go ahead.
Actually, I would have a question about the marine biodegradation results that you showed, because on that slide, it actually looked like that the PLA even behaved worse than the LDPE, and so I was wondering which literature data you used for this assessment, and did it also include other degradation processes like UV or like UV aging for this degradation that was going on there?
I've seen the same. I don't know, to be honest. So I'm now currently checking all the data that's from literature under which conditions it's measured, and if we can even put it in these slides. So for now, it's more preliminary results to see how we can use this model and which CO2 curves can we use. But I agree with you that it's an interesting result that this LDP is. I don't know. I will need to check in more detail.
But the basis was again CO2 curves that someone determined under some conditions. Okay.
Yeah.
Okay.
Yes.
All right. We also have one question from the chat from De Jong. He says, very interesting. Does your model take into account the three-dimensional structure?
No, not yet.
Okay.
But in the end, if you measure, or we hope at least that if you measure a CO2 curve on a three-dimensional structure, so not on the microplastics that you feed to the soil or to another environment, but to a 3D structure, that it will be, in the end, you get the same CO2 curve, so it should be suitable for three-dimensional structures as well. Do you understand what I'm trying to say? I don't know who the question came from, but
the question was for me. I mean, even in microplastics, I mean, it is really. I think it's influenced if it is more less a ball or a flat surface, so to say. I mean,
but that would mean that you would measure the how do you say, the CO2 evolution curves of different shapes of microplastics, and then you can feed that to the model and you can see the differences as well.
Exactly.
So in the end, our model works with any given CO2 evolution curve as long as it's measured in a specific environment under specific conditions.
Okay.
All right. Is there any more questions? Okay. If not, then thank you to Kai and to our speakers, Marieke and Patrizia. Thank you for the lovely presentations, and I think we can finish the meeting here.
Thank you.
Yes, Kai, go ahead.
Yeah, thanks, Dario, for taking here the moderation of this meeting. From my perspective, I wanted to thank the speakers to contribute here. And for me, one major takeaway today is the difference between persistent and non-persistent microplastics, which was shown experimentally by Patrizia what the difference is here, and by Marieke in the perspective of accumulation potential. And I think this is very important to look at this topic from this angle, and to move along from here. And to consider such kind of results and insights in the life cycle assessments and our assessments, what makes sense to use, what kind of plastics in which kind of application. So thanks very much. It was very insightful for me, and yeah, thanks.
Lovely. Absolutely agree. And thank you so much for the presentations. All right. If that's all, I hope everyone has a great day, and I hope you learned something good. All right. Bye-bye.
Thank you.
Thank you. Bye.